All right, so I'm going to talk a bit about there we go. I'm going to talk a bit about how maybe the most impactful way to have an impact in biology and in longevity is not directly in longevity, but through more data production and in biotech. So first of all, I'll go super high level here, but like biology is incredibly complicated. Death is very complicated. There's a KRAS G12D mutation. If a cancer patient was to get it, we don't really understand what is going on with the pathways. We don't really understand what is going on with how it defends cancer from therapies that we are normally used to for KRAS. And then it also has silent I always mess up that word, but micrometastasis in a way that we don't really understand what is going on, but we know that the survival rate is incredibly low. So we have some notion of the gene, we have some notion of the fact that it is very deadly and what it's doing, but when it comes to the actual molecular interactions that is causing the survival rate to be below 2%, we don't really have the tools to know what is happening. And there's an array of tools that would help us do that, but biology currently, they don't exist. Talking about the actual longevity, epigenetic drift, what many of us would consider like real kind of aging research. If we think about a 72-year-old man with clonal hematopoiesis, sorry, English second language, but you can think like we understand many of the high-level things that are happening, and we understand that their vaccine responses are going to be blunted. We understand that they're more likely to get leukemia. We understand that they are going to have problems with their, whether stem cells are going to work. But at the end of the day, we don't really understand which methylation sites drive dysfunction, how it interacts with metabolic signals, and whether partial reprogramming would help this patient. And all of this to say is not to focus on KRAS, not to focus on epigenetic drift, but rather it is incredibly complicated. Biology is very complicated, and we don't really understand what is going on at the micromolecular level, which leads to a lot of deficiencies in how we can address those problems. And so long term, if we think about a 20-year timeline, I think there's kind of two ways that we can approach this, or that we will have to approach this. One of them, I used to think, is kind of one-to-one digital twin of human biology. If we're able to better read to the molecular level what is happening at every atom to atom, and we're able to simulate that in an environment where we can test millions of cells, millions of therapies at the same time, then you can imagine we might be able to just do that instead of testing on humans, instead of testing on mice, and just advance therapies much faster. Nowadays, I actually don't think we'll ever get there. With all the advances in AI, with all the advances in data generation, I think we can get to a point where we still don't understand what is going on at such a complex level. Again, biology is not really meant to be human understandable. It wasn't really a constraint, such as computer science. When you think about computer science, obviously one of the constraints on how we develop ideas and how mimetic they are is how good they are and how much they develop. So it might be 10% less efficient, but if it's 2x as understandable by human, that idea will actually go further in computer science history. Biology didn't really have that constraint in evolution. And so when you think about biology, they didn't really care if a human could grasp it. And so we might think that if we generate enough data, it can approximate it through AI and then do that same function I was talking about, but without doing a one-to-one digital twin, at least not one digital twin that humans could understand. And so all of that goes to say that my argument is that working on better data and better ways to capture data on the lab, whether, and we'll talk about the methods themselves, but working on this better data might be the best way to work on biology and work on longevity by better understanding the human system. I wanted to cover 4 areas of biodata production that I find really interesting. NGS, whole genome sequencing, single-cell omics, spatial biology, and then protein sequencing. Obviously, these are way over-indexed on, but I think they make good benchmarks for where the field is at. And then what it enables, if we were to 10x the technologies over the next few years, what it enables for biology and what it enables for longevity research. So first of all, NGS, as everyone knows, 2015, it used to cost $1,500 to sequence a single genome. That machine is actually way bigger than this one. It's also, you needed 10 machines to really get the price to that. 10 years later, everyone knows this example, $100 cheaper long reads, methylation sequencing, ATAC-seq. If we're able to make that same progress over the next 10 years, you can imagine having a constant epigenetic clock for a person, for a group of millions of people, where we could get the human genome to $20. We could constantly be seeing different populations and how their epigenetic clock is drifting and affecting, and then do it also at a single cell level. And this is another area I'm super excited for. So you have 2015, $1 per cell, and really we could only do a few hundred cells. Like, this is a field to watch, in my opinion. Like, I'll show some graphs, but it's pretty insane how we've advanced from $1 per cell to now doing what, like 5 million cells with Scales Kit, with Parse Bio's kit. Obviously, 10x keeps driving the price down. And then if we can get to a point where we can do these aging atlases at the single cell resolution, where we can do clonal drift detection. So for the patient I mentioned earlier, if we could very early on identify that G12D mutation and see where it's coming from and how it's affecting and how it's further getting to the point where it becomes that deadly, that would be very informative. And just like a few more things on this space. So this is a field, this graph I like out of the design, but you can see, like, from 2010, we've gone from 10 cells to now 100 million cells within a data set. Hopefully everyone's familiar with the Tahoe data set that came out earlier. This is another one. So that's transistors since 1993 in a single chip. And then this is single cells in a single kind of protocol. And so you can see that's a log graph. And so you can see this is growing insanely fast. There's a lot of details that could be better in this graph, but the point is single cells is very exciting, and it's going to continue growing, and working on this technology is very, very exciting. Spatial biology, 2015, we could barely do a couple probes. We could do a couple proteins per tissue. Nowadays, Xenium and Cosmx from Bruker are both battling to get 20,000 gene panels. And then on the other side, we have unbiased like Curio, we have unbiased like Stomix, and they're trying to do at the unbiased level of all panels. Now, there's two problems there. So on the spatial imaging side, so when you do it based on images, we don't really have unbiased. And so we can't really get the location or the image of the sample. We need to, like, have the probes for the human. And then on the other hand, for the NGS-based spatial, it's not really single cell. So you have, like, multiple cells going into, like, a single probe. And so that is a huge challenge to work on that will really allow us to have a Google Maps of the organs and see how aging is happening in those organs. I won't go into protein sequencing because time is running out, but also a very exciting field. But the point is that if we can 10x tool capabilities, we can really, really advance the space, both of biology and of longevity, from early detection, precision treatment, rapid testing, but also the combined multi-omic tool set that gives us might be the most impactful way today to drive our understanding of biology. And then one day, whether it is through an oracle or whether it's through a digital twin, get to the point where we turn biology not from a science, but to an engineering practice where we can have that true impact that we all want to have. Thank you.
Give it up. Thank you very much.